Hello and welcome back, everyone. Sorry about the technical difficulties that we were having a while back, but glad I got it sorted. So, quickly, I would like to introduce Cell Modeler, which is a GPU accelerated multicellular modeling framework. Since this is a DeSci ideathon, so it's essential that we try to run a scientific simulation using cell modeler as an example. So it's a very good example of uh, understanding how we can run scientific experiments and computations in a decentralized manner. So cell modeler is basically a modeling system for uh, s- like bacterial cells, large scale multicellular systems such as biofilms, plant and animal tissue. So we have like developed models of cellular biophysics, gene regulation, and other intracellular processes, intercellular signaling. My work had been mostly on biophysics, working on the biophysics module for the past many years, since 2016. So the idea is to create a system where we can simulate the growth of the show the division of cells and everything, and so that the advantage here is in the wet lab it takes many days to do such kind of things, but when we try to do it in a computational manner, so we are able to uh, estimate the growth in a much faster time. So we can do it in a lot faster way. So that is the advantage of using GPUs, because we are able to simulate them at a much faster rate. So you can go through the GitHub and documentation also. Uh, Here in the GitHub we have really good tutorials. So you can see there are tutorials on biophysics, intracellular chemical dynamics, intercellular signaling and diffusion. So here there are a set of very good explainers that we can understand. So let me show you the first example, because those are the basics. So here we have the tutorial is on simulating cells on a single surface. So tutorial 1a.py, it's a Python program basically. So you can understand it's a Python library, so that you can use to run the simulations. So if you want, I can show you the Python file also briefly. So you can see here we have used cell modeler as a library. Here we are calling these modules, and then we are trying to simulate the cells in a programmatic manner. So when we do this, we need the GPU so that the work is done efficiently and quickly we can see the computation of cells in a like in periodic manner. So after this example, we also have some small example of divide called. So we are calling this divide because this is simulating the division of the cell, and there are also many parameters such as target volume and the volume. So if it reaches the target volume, then we can program the cell to divide. So we are setting the divide flag as true, so then only the cell is dividing. So there are a lot of mathematical uh, derived mathematical formula based on mathematical biology that we are incorporating in this module, so incorporating in these programs. So these are all the this is what essentially you can call computational biology, where it's a interdisciplinary way of looking into how life works. So uh, cells are very unpredictable. So my work has been to understand how Staphylococcus aureus essentially grows. So there are many diseases related to this particular bacteria. So initially I started working by simulating if Escherichia coli, or we call it E. coli. So we have three kinds of cells. So there are round shaped cells, there are medium shaped cells, and there are long shaped cells. So initially we started working on the round shaped ones. We used to work on the E. coli species of bacteria, because a lot of work has been already done on that area, and based on that we were able to build upon that, and so that we can uh, use those observations to understand Staphylococcus aureus, which is the other species of bacteria. Very mysterious. Like a lot of researchers will agree that it's a very mysterious kind of a species, like based on how they grow and how they behave in the like microcolonies. So this is one example of growing the cells in a cuboidal structure. So like as we is mentioned here, running this model for a while yields a clump of cells trapped in a box. So it's a really interesting way to for computer science researchers, computer scientists to be involved in a biological aspect of like biological application, where it's a very beautiful way of how computer scientists can uh, help in understanding biology. So it's a very essential thing that biologists and computer scientists work together, so that we can find solutions more easily. And of course, DeSci makes that very easy, very possible, because there are a lot of barriers are like removed when we try to do these kind of interdisciplinary research. So coming to the running this on different platforms. So when we try to run it, we essentially use all those centralized based architectures. So institutions are running machines in their labs, and there are these machines are all having their own hardware, and we have to run it in a node by node manner. So when it comes to NuNet, if we relate this to NuNet, we are able to run it in a decentralized manner. So we have the service provider dashboard, where we can run any program here, like any Python program. If it needs a dependency, we can specify it here. So we use the pip or the conda package manager to install the particular dependency, and then we can run the programs. So here I have mentioned a small example that uses cell modeler to run a simulation. So let me go through this program first, so that it will be easier for you to understand what actually we are trying to do here. So first we are cloning the repository that I just shown, this particular repository. We are cloning it, and then we download a few custom files that I've saved as a snippet on GitLab, because I need some modifications to do to the program, so that I can run exactly what I'm trying to do. And then I'm using a tutorial based example. So this is a tutorial file that I showed you, the tutorial A or tutorial 1A, 2A. So I picked tutorial 2A here, 2A, and I'm trying to run it on this platform. So here I have specified this link, and it is going to do all of these things. Yeah. So, and also I'd be honest that generative AI has already also been very helpful in scripting these things and making things a lot easier. So I would also recommend all the viewers to adopt generative AI as much as possible, because it makes coding really easy, and you can it reduces the gap between the researchers, so that they can work on some problem. So based on this script, we can move ahead and try to see whether it can run this simulation or not. So since Cell Modeler is using PyOpenCL, as you can see in this particular documentation, Yeah, you can see it leverages CPU and GPU parallelism via OpenCL and PyOpenCL. So so then we of course need the PyOpenCL platform. So that will be the preferred platform that we would be choosing instead of TensorFlow or PyTorch, because TensorFlow and PyTorch are definitely there, but they are for the machine learning based job request. But since we have the PyOpenCL, it becomes very ideal for running this kind of simulations. So when we select PyOpenCL, we do not need to install any dependencies, because I've already specified in the script here what to do and what not to do. So after so we can remove this here. We just wanted to show you because it's uh, we can we can, if you need a dependency, I wanted to show you so how to add it. So then we can give it a particular time frame within which I expect this simulation to complete. I give it a maximum time limit of 30 minutes. So then we can uh, try to move ahead with the simulation. So this is the tokenomics part, where we specify what amount of tokens I want to give it to run the simulation. So I'm giving it 1 NTX here. And then you can see we're using the blockchain Cardano here. And we also have a resume job feature that Santosh has been working on. Very interesting. Yeah. So in case this is like we ran the simulation for some time and we don't want to lose that progress, we can resume that job. But uh, right now, let's try to focus on the simulation. So yeah. So here we see all the list of the compute providers. So we can see which machine has a GPU, which doesn't have a GPU. So it depends on, this is a test net. So the more GPUs you onboard here, more GPU machines, you will be seeing this GPU as yes. So here we have one particular GPU. Uh, yeah. So there's a Tesla P100 16 GB, and there's a 1050 Ti also here, I think. So let's try this on this Tesla P100. So I think we could try this one also. We could, but let's see what happens here. If it doesn't work, then we could try again. So this is a experimental demo. So we are all experimenting. So let's try this. Okay, so job has been submitted. Okay, so it failed to get the GPU info. So let's try some other GPU then. So let's try this one. Sorry. So I think we are facing some difficulties here, but we have run a simulation previously, but right now we are facing some issues with it. Okay, could try some. What happens if we do it on a CPU? Let's see if we try to do it on a CPU, would it run? You see, it took the request. So the job has been submitted. So this is essentially what happens. Like when you submit a job, it will try to run. It will try to download the particular container, the PyOpenCL container, onto the machine, and then it will try to execute the job over there. So since we have time now, we can try to work on this further. But it's unfortunate that we couldn't show it to you on the first attempt. So let's keep trying since we have the time now.
Actually, before the container is downloaded and all,
Yeah,
like
yeah.
there is some blockchain.
Yeah, but yeah, the blockchain part we removed because we wanted to show the demo for quickly showing the demo. Yeah, you mean the wallet, right? The
Yeah,
wallet.
yeah.
Yeah, yeah. So you can try to show. Try to share the screen on the terminal. Let me share this terminal here to see the number of peers that are available. Yeah. So in this, let's see this is the NuNet peer list command that we can list all the machines. So I see we cannot currently see. Actually, I was trying to. I was expecting to see my lab machine. So this particular peer ID is of one of my machines in my lab, and there's also another machine. This is this particular machine. Both are having GPUs. So it because since we just onboarded, there's some time it would take to reflect on the DHT peers. So these are the peers where we can request the jobs. So yeah. Meanwhile, let's check the progress on the. I checked the progress on the job that is submitted. It is still under deployment. So, you have any questions on Cell Modeler?
I think we don't have
Okay.
biology people today.
No, it's like you can ask any general questions if you want. But meanwhile, I can show you the screenshots of the experiments that we did on this interface. So just give me some few moments. I'll just show you the uh, screenshots.
So while modeling the cells,
Yes.
what are the basic conditions and parameters you choose, and like and what is the rationale behind it? Like when you
Yeah.
trying to create that model, if you can.
Okay. So parameters by yeah. So since you mentioned parameters, that is one of the most important aspects because unless we have these parameters, we cannot begin the experiment of the simulation. So we have to specify the length of the cells that we are going to see divide. So based on these lengths, we can the other cells are also going to have the same sizes. So these are one of the parameters. So there are many more parameters also that you can when you try to run the simulation, you can set those particular simulation parameters, and it will use only those parameters when the simulation will start, like the growth of the cells would be visible.
Right.
Yeah.
So so what is the end goal of the simulation to see how the cells mutate over time?
Yes,
And
the yeah. So one of the most essential things is like you can compare it to the wet lab results, because on the wet lab it takes weeks and even months to get a complete
right.
uh, culture of the bacteria.
Yes, it
But
expedites the whole process.
yes, yes. So we can see it in a much earlier. Yeah. So I had anticipated this issues could happen, so that's why I had bookmarked all those screenshots that we already have done it successfully, so we can show you that because we have done it on both NVIDIA and as well as AMD. It's I think we should have shown you on the first day itself, because on the first day we were able to run the jobs from this venue. So my machines were visible at the lab, and not only that, we were able to run it on a random machine also from this venue. Yeah, I'll share. I'm going to share here. So yeah, so here you can see in the outcome, like this is the same cell modeler script that we were trying to run. So after the simulation finished, it will. I'm not visible. I'm sorry. Okay, there's some issue with the screen sharing. Let me try it again.
Yeah.
Yeah, I think is it clear?
Yeah.
Okay. So this is the particular uh, outcome that I was trying to show you. So here a 3060 Ti has been used, and this is the same tutorial 2A that we showed you in the cell modeler documentation. So from there it starts from 2 cells and then keeps dividing. It's a pretty remarkable thing I discovered, that we can run cell modeler experiments on NuNet. Not only that, I have experimented with DeepChem also, that Stanley had mentioned in one of his talks earlier, and it's fascinating that we can explore that as an area as well, because it is related to machine learning also. So DeepChem uses TensorFlow to run the jobs. So we can try to use the TensorFlow. We can select the TensorFlow platform and then try to install DeepChem on that. So I'll quickly show you the DeepChem page also. Just hold on. Deepchem.io. Yeah. So here also you can see, if we look into the installation part, you can install it with pip and cell modeler. Sorry, in the DeepChem would be installed on the interface. So we have Docker also, Conda also, and pip also. So we provide on NuNet SPD, the service provider dashboard, we provide both pip and Conda. So you can use either of these platforms to install DeepChem. So we already have. So this is a Conda channel, and we use Mamba, which is a drop-in replacement of Conda that makes it really fast to install the packages. Because if you see natively Conda, it might be a bit slower. There might be latencies when installing the dependencies through Conda, but when you install Mamba, it installs it really quickly. So we already have this Conda 4G channel added to the cell modelers I'm sorry added to the uh, SPD interface. So when it downloads the OpenCL or TensorFlow or PyTorch containers, you can already have these kind of channels available. So in the OpenCL containers specifically, we have these channels available. We have a lot of scientific channels available. So you just have to mention the package name, and the dependencies would be installed. So you can install this as a dependency on the SPD. So you just have to copy this and like paste it on the SPD interface. So here you have to select the dependency and you can just paste it here. So when you paste it like this, you can yeah, I'm sorry, part of 2.7.1. So you can just specify this here. You select TensorFlow and you can run any particular program on DeepCam. So this program can be something that is running a DeepCam simulation. So if we visit the DeepCam homepage, you will find the GitHub is also present here. You can just directly go to the GitHub and see the examples. So through these examples, you can understand what kind of programs you need to write so that you can run these simulations. Yeah, so you can use any of these one. So there are a lot of examples here. So there's a benchmark file also. We can try this one. So you could try to benchmark this using the SPD. So you just need to copy the raw URL and you just copy it and you can try to paste it here and then you can try to run this one. So let's try randomly. So we are seeing all those machines here. Okay, I think that particular machine is having some issues. So let's see what happened to that CPU request that we gave. There was a CPU request that we gave, right? Yeah, it's still submitted. There's some issue with it, I think. So let's try that again. So, okay. No peers found. It's pretty unfortunate. Let's try to restart the DMS, or shall we try to restart the DMS? Oh, sorry. So this is what happens when you do the testing. It runs, and then in the live demo, it's not working.
Biggest nightmare.
Yeah. So just try to restart the DMS. No, so it's great. Yeah. Yeah, yeah, it's there. Let's see. It's fine. It's fine. So, I think there's some issue with the camera. Check. Let's try to see the peers now. The same peers are coming. Unfortunately, my machines are also not coming here. So these two, I was expecting to see these two. Yes, but it's not showing right now. So let me try it from my home PC. Let's see.
Someone has to accept the request.
Which one?
And it is
It's already pre-configured. Some issue over there, I think. You can run simulations on CPUs also, but just for testing purpose, I'm just checking whether it's able to deploy. I need to call Ola, I think. I need to call Ola.
Sam also has a GPU machine.
Yeah, I think that's Sam's machine only. Yeah, I think that's Sam's machine.
had a backup.
You can want to do it.
No, like I had a backup in my top.
Yeah.
What happened to you? You want me to like how does it work?
Tested it multiple times, but it's now itself that it decided not to work. How to you can send a message to this peer and check the documentation. It's okay. I think there is some issue with the bootstrap peers maybe. So
You can check the logs.
yeah.
Have to restart.
Okay, I think we can take a short break now.
Yeah,
Yeah.
I